Hi and welcome to my OCR A-level biology revision session with me, Christine. So today's lesson I want to look at ultrafiltration, which is part of your excretion topic in module 5. So just remind ourselves of the structure of the kidney nephron, which is made up of your glomerulus, your Bowman's capsule, your proximal convoluted tubule, then going down the loop of Henle, up the distal convoluted tubule, to the collecting duct and down in the medulla. It will then lead into that pelvis. So we're really interested right now because we're looking at ultrafiltration. We want to look at the glomerulus and the Bowman's capsule. And what they love to do in the exam is they love to give you the histology pictures without obviously the labels. So if they give you a picture and they ask you to identify the Bowman's capsule, it's really clearly identifiable in this section, which is clear, which doesn't have any staining, and it looks like a C shape. So it's like a cup. So you always want to identify where are the Bowman's capsules. That's the region where you don't see the staining. Now, the other part, the glomerulus, this bundle of capillaries, is very identifiable. So what we want to do is we want to have a look at this a bit more so that we can understand the process of ultrafiltration. So ultrafiltration is this filtering of all of the small molecules out of the blood plasma. Now, when we say small molecules, we're talking about our water, our salt, our mineral ions, our sodiums, our potassiums, urea, glucose, and amino acids. So the filtering that happens at the tissue fluid is very similar to what happens at the kidneys, except all of it is going to be squeezed out of the blood and into the kidney nephron, which could then lead down to be removed from the body. So how does it happen? Well, the key thing that's happening is we need to understand that we have blood that is flowing into the glomerulus, and the blood is flowing in through what's known as an afferent arteriole. We've got our glomerulus, our bundle of capillaries, and the blood's going to flow in through the glomerulus and out through the efferent arteriole. So it's really important we look at the size of the lumen in the afferent and the efferent. So the afferent arteriole is slightly wider, and what that means is it's flowing more blood into the glomerulus, and therefore less blood is flowing out, and that creates a high hydrostatic pressure. And that high hydrostatic pressure is going to allow for these small molecules to be squeezed out and into the Bowman's capsule. Well, let's just have another look and zoom a bit in further. So we've got our glomerulus, we've got our Bowman's capsule, and we know that the glomerulus is this blood capillary, this bundle of blood capillaries. Well, we know that that high hydrostatic pressure in the glomerulus is going to force the blood through fenestrations between the cells. So those fenestrations between our squamous endothelial cells is going to allow for the blood to move through, but it acts as a bit of a sieve. What it's doing is it's going to stop large molecules from getting through. Well, it's not just about the fenestrations. It's important that we note that there's also what's known as a basement membrane. So a basement membrane is going to prevent these large molecules, these proteins, and also our erythrocytes and our blood cells from squeezing through into the Bowman's capsule. So what we're doing is we're filtering, which is why it's called ultrafiltration. We're filtering because we're only allowing small molecules through. So what we also have is we have these extra cells that are differentiated cells called podocytes. Now, these podocytes form an additional filter. They're ensuring that if anything has got through the fenestrations, has passed the basement membrane, what they're going to do is they're going to act like a further sieve and prevent any of those large molecules from getting into the nephric tubule. We don't want to lose any of these large molecules. We don't want to excrete them. They should be maintained within our blood plasma. So this is another form of a barrier. So we can tell if there's any form of blood or large proteins found in our urine, that indicates a problem in our kidneys at the glomerulus and the Bowman's capsule. So I hope you've liked this video. If you have done, then please do click on the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com.